Hello, my name is Chris and I teach chemistry. I've been teaching chemistry for over 25 years, ranging in all levels from AP chemistry down through conceptual chemistry. During the past 5 years, I have also been working extensively with tutoring and home instruction, again, with every level of high school education from AP through conceptual. In my home instruction and tutoring, I have also helped students with biology, anatomy and physiology, environmental science, and physics. I have degrees from Cornell University, from the University of Maryland, and I have several teaching certificates from Rutgers University. I have been an AP reader. AP readers are the individuals who help score the AP exams, and my classes have been involved in piloting the AP chemistry lab program that was generated by the College Board. Today, I would like to share with you an introduction to photoemission spectroscopy. Photoemission spectroscopy is a technique that goes under several names. Sometimes it's called PEST for its initials, and sometimes it's called photoelectron spectroscopy. They all mean the same thing. Photoemission spectroscopy is an analytical technique. From an AP perspective, it's used to determine the electron configuration and identity of elements. From an advanced chemistry perspective, it can be used to determine bond type in different materials. The AP program has 4 required skills associated with photoemission spectroscopy. Today, we are only going to deal with 2 of those. I'd like you to understand how a PEST spectrum is generated, and I'd like you to be able to interpret that spectrum to identify the elements associated with it. In order to understand this topic, you should have a basic understanding of the photoelectric effect, and you should be able to understand what an electron configuration is and how it is related to atomic structure. If you're not familiar with those topics, you might want to stop this presentation and go back and review those topics. This is a schematic of how photoemission spectroscopy works. In this scenario, there is a source of photons, typically in the ultraviolet or X-ray range. The energy associated with those photons is known and identified. Those photons are directed at a sample, and when they hit the sample, they cause electrons to be ejected. The electrons that are ejected are collected by an energy analyzer. The electrons that are ejected have less energy than the photons that hit them because it takes energy to pull the electrons out of the atom. This energy is known as binding energy, or it's also known as ionization energy. They're slightly different terms, but from the perspective of this lesson, they mean the same thing. Once again, high energy photons strike a sample, electrons are ejected, and those electrons are measured for the energy that they contain. The product of this technique is a graph. The graph is called a PEST spectrum. On one axis, it indicates the ionization energy or binding energy associated with different electrons. And on the other axis, it shows the relative numbers of electrons that have those energies. It's typical for a PEST spectrum to have discontinuities in it or breaks within the scale because the binding energies of electrons varies hugely along inside an atom. It's important to note which end of this axis has electrons with the highest energy. Depending on who prepares the graph, they can be on the left or they can be on the right. In this graph, the electrons with the highest binding energy are on the left side of the axis. The electrons with the greatest binding energy are those that are closest to the nucleus. So if you think back to electron configuration, this peak would represent electrons in the 1s sublevel. As we move further and further to the right on this axis, we encounter electrons that are further and further from the nucleus of the atom. The sequence of these peaks follows a typical electron configuration sequence. So this peak would represent the 1s sublevel, this is the 2s sublevel, this is the 2p, and this is the 3s. The heights of these peaks represents the number of electrons found in those sublevels. We know that this first peak holds 2 electrons, and the reason for that is that the Aufbau principle says we must fill the 1s sublevel before we begin filling the 2s sublevel, and the 1s sublevel holds 2 electrons. So this peak, the one at 9.07 and the one at 0.74, each hold 2 electrons. The peak at 5.31 megajoules per mole is 3 times higher than the other peaks. That means it holds 3 times the electrons for a total of 6. The electron configuration that this graph represents is 1s2, 2s2, 2p6, 3s2. Once we know that electron configuration, we can determine that this graph represents the element magnesium, because magnesium has 12 electrons in its neutral state. Here is another PES spectrum. Again, the highest energy, the electrons with highest binding energy are in the 1s sublevel. This is the 2s, the 2p, 3s, 3p, and 4s. We have 2 electrons in these medium-sized peaks, 6 in the tallest, and this peak is half the height of the other s sublevels, so it has just 1 electron. This would be 1s2, 2s2, 2p6, 3s2, 3p6, and 4s1. This element, this configuration represents the element potassium. Our final PES spectrum has 5 peaks in it. The highest energy peak represents the electrons closest to the nucleus. This would be the 1s sublevel, the 2s sublevel, the 2p, the 3s, and the 3p. In this case, the peak at 1.76 megajoules per mole is 1.5 times taller than the S peak, which means that it has 1.5 times the number of electrons, or 3. So this configuration would be 1s2, 2s2, 2p6, 3s2, 3p3, which represents the element phosphorus. With this very brief lesson, I would hope that you might be able to describe how a PES spectrum is generated. Once again, there are high energy photons that strike a sample, causing electrons to be ejected. The energy associated with the ejected electrons is interpreted to find the binding energy of each of those electrons. I hope that I would like you to be able to interpret a PES spectrum to identify the electron configuration of the represented elements and to produce the identity, to generate the identity of the element represented. Thank you for being part of this lesson today. I hope you learned some chemistry. I look very much forward to working with you in the future, and I hope you have a lovely day. Bye-bye.